in the following slides, we can see that yes. So in the next slides, we will see that how this R can be varied from one molecule to other molecule. But this R can be 20 different types of R. Side chains can be present over there. Okay.
Hello,
So,
sir.
yes.
Excuse
Yes.
me.
Yes, Milon
Actually,
sir.
live streaming is now on, so you may proceed.
Okay. Okay. Thank you. Thank you. Yes. So in this amino acid structure, as I told you that these four central carbon atom is there, one amino group, one carboxylic group, and one R group is present. Okay. So moving on to the next slide, these, as I told you in the previous slide, that 20 different types of molecules or R groups can be present in the amino acid. Here actually we are showing that 20 amino acids. Okay. And these 20 amino acids can be differentiated or divided into different groups. This I have taken from a book, but when I usually teach to my students, I usually tell them how to remind these 20 amino acids very easily, the names as well as their structure. Okay, five amino acids are very simple amino acids which can be said very quickly. And the very beginning of the list, that glycine, alanine, valine, leucine, and isoleucine. These five are the basic, and not basic, these five are the very simple amino acids. After that, we can say that two acidic amino acids are there, which are the aspartic acid and glutamic acid. Now you see, if the acidic amino acid is there, acidic amino acid means there are an extra acid group in the R portion. So if the acidic amino acids are there among the amino acids, then it must be some basic amino acids should also be there in the list. So in basic amino acid list, there are two different amino acids which are lysine and arginine. So already we are in the ninth position, 9 amino acid we have done. Okay, so simple amino acid has gone, then acidic amino acid has gone, then basic amino acid has gone. Then we will go to the amines. That means the amines of the acidic amino acid. Okay, that is aspartic, the amines of aspartic acid and glutamic acid. What are they? These are the asparagine and glutamine. So again, two amino acids are there, asparagine and glutamine. Then we have two alcoholic amino acids. That means some alcoholic group are present in the R group. That's what are they? That is the serine and threonine. Okay. And two amino acids are there which are actually sulfur containing amino acids. And these are cysteine and methionine. Okay. And three amino acids are there which are actually aromatic in nature. Hello, can you please mute everyone? Can you please mute everyone's mic? Can everyone please mute yourself? Yes. Everyone please mute yourself. Okay. So moving on to the our next discussion, that up to this we have come to 16, 17, 18. Okay. We are now having three aromatic amino acids: phenylalanine, tryptophan, and tyrosine. Okay, so 18 amino acids are already done. Two amino acids are left. These two are the complex amino acids. One is the proline, another is the histidine. Okay, so this way we can divide our amino acids, and my students can remember the 20 amino acids very easily. Okay, so now moving on to the peptide bonds. How the peptide bonds are actually found. Say, for example, these are three amino acids: amino acid 1, amino acid 2, and amino acid 3. These peptide bonds are actually needed to form a polypeptide. That means the protein molecules. These amino acids, as I told you, these are the building blocks. These are the units of the protein, but they have to be together. They have to bound together to form a protein molecule. As bricks are the unit for a building, but if you just put the bricks one after one or one over one, building cannot be formed. Okay, you have to cementing them. You have to put some cement between them to bridge the bricks. Just like that, you have to have some bond between the amino acids so that the protein can be formed. So amino acid 1, if amino acid 1, amino acid 2, and amino acid 3 are there, then these just follow these red mark. Okay, the H from the NH2 group and OH from the COOH group, they will club together and water molecule will be released by making a bond between this carbon atom with this nitrogen atom. Okay, so this carbon atom and this nitrogen atom will make a bond, and that bond is known as the peptide bond. So likewise, all the amino acids will be bonded together to form a peptide bond. This is also the same way the peptide bond will be formed, and amino acid 1, amino acid 2, and amino acid 3 will be bounded together, and many amino acids will can bond one after another. And in this way, you can see that, say for example, these three amino acids are forming one protein, then in that protein, amino acid, in case of amino acid A, the NH2 group is free. It is not bounded with any other COH group. And in case of amino acid 3, the COOH group is free and it is not bounded with any other NH2 group. So, in a polymer of these amino acids, which are known as a polypeptide, as many numbers of peptide bonds are present, for that reason it is known as polypeptide. In one polypeptide, one nitrogen end is there and one carbon end is there. The end where the COH group is present, that is known as the carboxyl end, and the end where the NH2 group is present, it is known as the amino end. Okay, so this is the polymer of an amino acid, of all the amino acids, and this, in this way, the amino acids get together to form a polypeptide or protein molecule. So this is actually just the structure. Protein has a different level of structure, one after one. If we just add the amino acids in a chain, then it will be a polymer of the amino acids, which is known as the polypeptide. And the just chain of the polypeptide is known as the primary structure of the protein. Okay, there is no separate structure, only a chain of amino acids. So that's it. Then these amino acids will fold in such a way that it can have some second degree of structure, which is known as the secondary structure. We will discuss about the secondary structures of the protein molecule. After that secondary structure, I'm just, for example, I'm telling that alpha helix, beta sheets, these are the secondary structure of the protein molecules. Okay. Now, maybe two alpha helix and three beta sheets, they will club together to form a tertiary structure. Okay. So this is kind of tertiary structure. And in some proteins, these tertiary structure is the active molecule. They can act directly, or else in some cases, more than one tertiary structures or the units will clump together to form an active protein. That is known as a quaternary structure. If we divide or if we break the quaternary structure, then we can have more than one these tertiary structure. Okay, tertiary structure proteins are the separate units. So if we add two or more than two units together to form its active form, to make its, active form, to make its activity, to perform its activity, then it is known as the quaternary structure. All the proteins might not have its quaternary level of structure. Okay, few proteins may act at its tertiary structure also, but few proteins may act at its quaternary structure level. So these are the four levels of structure of the protein molecule. Now, as I am telling that there are several types of secondary structure, these are the most important structure of the protein. Alpha helix is the very important secondary structure which is present in the protein molecule. You see, this is a helical kind of structure. Okay, you maybe heard about the DNA helix. So in the protein also there is a helix, which is known as the alpha helix. It is a single helix. Sorry, it is a single helix. Okay, and why this helical structure has been formed? You see, these are the hydrogen bonds. The black dots are the hydrogen bonds. So if one amino acid can form a hydrogen bond with a second amino in a subsequent amino acid, then this helical structure can be formed. And how this amino acid can bind with one another by this hydrogen bond? There is a law. Okay, the first amino acid should be bound with a hydrogen bond with the fourth amino acid, and the second will be bound in the fifth, and the third will bound in the sixth. Likewise, if the hydrogen bond is formed, as I told you that all the amino acids has two groups, One is the amino group, the other one is the carboxyl group. So H from the amino acid, O from the carboxyl, they can have bond by the hydrogen bond. So the NH group from one amino acid to the COH group of the other amino acid can form the hydrogen bond. Okay, although they have already been interacted for the peptide bond, but C double bonded O and one NH is all present there. Okay, still there to make this hydrogen bond. They are not involved in the peptide bond, so they are free as it is. So they can form this hydrogen bond. So one atom, one molecule of amino acid, the first amino acid will make a hydrogen bond with the fourth one, second will make a hydrogen bond with the fifth one. Likewise, it will go. If the hydrogen bonding pattern is like this, then it will make a helical structure, which is known as the alpha helix. Second most important secondary structure in the protein molecule is the beta sheet. Beta sheet is just like, I can give the example, sorry, that in the beta sheet, what happens, the secondary structure, um, I think all of you have seen the asbestos sheet, what we are used to cover our houses, right? So how it looks like, the beta sheet is actually looks like that. Okay. So this is the one polypeptide chain, and this is another polypeptide chain, and among them there are some hydrogen bonds again. These hydrogen bonds are actually very much important to make this secondary structure in the protein molecule. In the beta peptide sheet, it can have two different morphologies. One is the antiparallel, the other one is the parallel. If the protein amino acid chains goes like this is the amino acid chain going on. Okay, here one beta peptide sheet, again it is one beta peptide sheet, again it is one beta peptide sheet. If it goes like this, then it is known as antiparallel. Why? Because they are parallel, but they are moving to, I mean, in an opposite direction to each other. For that reason, these are known as the antiparallel beta sheet. And if the sheet is going like this is the arrow is the beta peptide sheet. Then again some amino acid chain is there, and then again this beta peptide sheet is there, and another amino acid chain, and again a beta peptide sheet in the same direction. If the beta peptide sheets are moving, then it is known as the parallel beta sheets. Okay, so beta sheets can be of two types. One is the antiparallel, the other one is the parallel beta sheet. Next is the triple helix. It is not very common, but it is there in the secondary structure. Keratin, by which our hairs are formed, they have this kind of structure, triple helical structure. The three polypeptide chains actually coiled together to form a triple helix. Okay, and random coil is also there. Random coil means nothing, no specific formation of a secondary structure. If the polypeptide chain is formed randomly coiled, it is not following the rule of beta sheet, or it is not following the rule of alpha sheet, and it is neither, involved in the triple helix, then it will form just a coil like this. These portions, it is a randomly coiled. These green arrows are the beta sheets, and this purple, feather-like structure, this is the alpha helix, and the rest of the portion is just randomly coiled. You are seeing to pack the protein. So these are the random coils. Okay, in this photograph, I am showing that if this is the protein molecule, then this portion will be the alpha helix, this portion is the beta sheet, and the other portions which are just randomly coiled, these are known as the random coil. So these three actually are the major part of the protein secondary structure. Okay, so now this is a very, small pathway, just in a very overview kind of thing I am showing. This pathway will be discussed in a very detail tomorrow, maybe, or day after tomorrow. I forgot the schedule. Professor Thakur Chaudhary is going to talk about this pathway. I'm just showing you in a nutshell that how the, protein is formed. It is coming from the DNA molecule. Later on, I'm going to show you the DNA structure, but going before that, and just telling that three major steps are involved in the formation of the protein. First, the replication, that means the DNA dependent DNA synthesis. DNA will synthesize by its own. This is actually not, related to the protein synthesis. For the protein synthesis, next two steps are important. The transcription. From the DNA, RNA will be transcribed. All types of RNA in the cell will be transcribed from the DNA itself. And from the DNA, the protein will be formed. Okay? Replication, transcription, and translation. Synthesis of DNA by its own, synthesis of RNA by the DNA, and synthesis of the protein by RNA. This is the central dogma of molecular biology. In this pathway, the protein molecule has been synthesized in the cell. The detail will be talked about and talked by Professor Thakur Chaudhary later on. Now we are moving on to our third molecule, third macromolecule, which is the nucleic acid. This is very important, and actually for nucleic acid, we all are similar as well as we all are different. This is a very, beautiful thing about the nucleic acid. We all are human. Okay, why we all are human? We all have two hands, two eyes, one nose. Okay, we have a head, we have 2 ears, we have two legs. We all are similar. All humans are similar. That is for this nucleic acid. And each and every human being is different. I am not looking like Professor Thakur Chaudhary. Professor Thakur Chaudhary is not looking like Professor Calcutta-wala. Professor Calcutta-wala is not looking like Professor Milan Bonik or Professor Atri Ghosh. So we all are differently looking. This is also because of the nucleic acid. So this is the most beautiful nature or property of nucleic acid. Each actually gives us the similarity with all other human beings as well as it gives us the differentiation with other human beings. So this is the main important and very beautiful nature of the nucleic acid molecule. Yeah, some noise is coming. Okay, yeah, now it's fine. So these are the nucleic acid molecules. As I told you earlier, that two major types of nucleic acid molecules are there. One is the ribonucleic acid, the other one is the deoxyribonucleic acid. Okay, ribonucleic acids are the RNA molecules, and deoxyribonucleic acids are the DNA molecules. Please mute yourself, everyone. Please mute yourself. Yes. So here I am going to discuss about these two macromolecules, the DNA and RNA. In DNA, the major difference between the DNA and RNA, I am going to talk about later on also, that the sugar molecules. As I told you, the ribose sugar, when I was talking about the sugar molecules, that ribose sugar we are going to find in the nucleic acid portion. The ribose sugar and the phosphate group, these two are the backbone of these nucleic acids. Okay, one sugar, one phosphate, one sugar, one phosphate. Likewise, it will go on, and it will form the backbone. And and it will form a helical structure, as we found in the alpha helix. It will also form a helical structure, the sugar and phosphate backbone. And the third component for the nucleic acid, the very important thing is that the nitrogenous bases. Okay, so nitrogenous bases are present in the sugar molecule, and it protrudes outwards inwards the sugar phosphate backbone. And one nitrogenous base will interact with the other nitrogenous base present in the opposite side of the sugar phosphate backbone, and it will form a hydrogen bond to make a double helical structure in DNA. Okay, and in RNA, as it is a single stranded structure, so there is no scope of hydrogen bond among one um, nitrogenous base with the other nitrogenous base of the next helical structure or the next helix, as there is only a single helix. So the DNA backbone, actually I told you, the composed of, this is the one nucleotide, this is another nucleotide, and here in the 2' position, sorry, 3' position, there is one OH group. Why it is known as the deoxyribose? I think you all know that in case of 2' position, there is no O group, no O. Okay, there is an only hydrogen atom. For that reason, this is known as the deoxy. Without oxy, deoxy means without oxygen. There no oxygen is present. For that reason, it is known as the deoxyribose sugar. But in case of the RNA molecule, there is there is an OH group present over here at the 2' position. For that reason, that is known as the ribonucleic acid or ribose sugar present in that. Okay. So here, this OH group and OH of the phosphoric acid group present in the. May I know, please, who is making this noise? STC sir, please, either mute him or just, remove him from the meeting. Is it clear? Is it now visible again? I'm sorry for the distraction. Is it clear?
It
Is
is
it?
clear. It is
Yeah.
clear, Rajat.
STC sir, please, take care who is
Oh,
making the
yeah.
noise. Okay.
Yes.
Yes. Thank you. Thanks a lot. Okay. So we are moving on. That this OH group of the phosphoric acid there, the OH from this group will interact with the H, and the water molecule will liberate it out by making one phosphoester bond. This is the acid group, this is the OH group, alcoholic group. So with the acid and OH alcohol, it will form an ester bond. And as it is the phosphate group, it is known as the phosphoester bond. Okay, so one phosphate will make two ester bonds, one with this OH, another with this OH. So it is known as the phosphodiester bond. So in a chain of the polymer of the DNA backbone, it will be the phosphodiester bond. One phosphate group will be bounded with two phosphoester bonds with the two adjacent sugar molecules to make the phosphodiester bond. And as I told you, the bases are present over here. These bases will be present at the 1' position, and they will interact with the other base for the next chain of the DNA in the next strand. Yeah, two strands in the DNA molecule. How they actually run? These two strands are running anti-parallelly. As I told you that in case of the beta sheets, two anti-parallel, two sheets are moving opposite direction, then it is known as the anti-parallel. In here also, two strands of the DNA molecule runs in opposite direction. That means in the previous slide, if I see that this is the 3' and this is the 5'. So when this bond will be formed, then the 3' OH again will be free without making any bond, and the 5' phosphate will be free without making any other bond. So as in the protein molecule, we had N-terminal and C-terminal. So here also we will have a 5' terminus and a 3' terminus. Okay. So likewise, we actually designate the this DNA or RNA chains.
Hello.
Here, yes. Okay. So here, in this anti-parallel direction, one chain is moving from 5' to 3' direction, and the other chain is complementary chain is moving from the 5' to 3' in the opposite direction. Likewise, it is going. So if we see this photograph, so here this is the 5' to 3', and here this is the 5' to 3'. Likewise, the two chains actually call together to form the double-stranded DNA molecule. These double-stranded DNA molecules are actually, DNA is usually a right-handed double helix. Okay, so this is the right-handed double helix. This is the left-handed double helix. In DNA, this is not present. The helix is not moving in this direction. In DNA, the helix is moving in this direction. Okay, so likewise, we will draw our DNA molecule. Position of the bases. Actually, bases are protrude inside the, in this sugar phosphate backbone chain. Okay, why? Because if they cannot protrude to each other, if they cannot face each other, they cannot form the hydrogen bond. For that reason, they are present inside the helical structure. If this is the helical structure, these, nitrogenous bases are present in between the helical structure to form the hydrogen bond to one another. Okay, and here always guanosine, four nitrogenous bases are present: adenosine, thymidine, cytidine, and guanosine. Two are the purines and two are the pyrimidines. Purines are adenosine and guanosine, and pyrimidines are cytidine and thymidine. In case of ribose sugar, that is the RNA, thymine is not present. Instead of thymine, uracil is present there as a nitrogenous base. And always one purine will pair or hydrogen bond with one another pyrimidine. Two pyrimidines cannot make any base pair. Two purines can also not make any base pairs. Okay. G is a purine, C is a pyrimidine. So G and C will make the base pair and they will have three hydrogen bonds. A purine, and T, another pyrimidine, they will make the hydrogen bonds between them and the number of hydrogen bonds are two. So GC will make three hydrogen bonds, AT will make two hydrogen bonds. Okay, likewise, the pairing will be done. Here I am just showing the detail of the hydrogen bonding. This is the thymine, this is adenosine, this is guanosine, and this is cytosine. You can see there are two probability to form the hydrogen bonds. For that reason, A will make two hydrogen bonds with T, and there are three probabilities for the hydrogen making the hydrogen bonds. For that reason, C will make three hydrogen bonds with guanosine C. What is the Chargaff rule? Chargaff rule is the rule behind this base pairing. Okay, as I told you that always A will form with T and G will form with C. Why A will form with T and G will form with C? The adenosine always make the hydrogen bond with T because as I told you that purine will always make a bond with pyrimidine to make the space well utilized. If two purine will make the hydrogen bond, then it will need more space. And if two pyrimidine will make the hydrogen bond, then less space is required. Then the double helical structure will be distorted. It will not be uniform. Somewhere it will be, bulged. Somewhere it will be squeezed. So it will be a distorted structure, but that can be done also. But why it is not possible? Because many protein molecules and many enzymes has to scroll over these DNA molecules. And protein and you know that protein and enzyme molecules are very specific to the structure of their substrates. Okay, they don't have eyes, they don't have nose, they just recognize their substrates by their complementary structure. So if the structure is not uniform or equal throughout the DNA chain, then the protein cannot scroll throughout the chain. For that reason, the chain must have a uniform structure throughout. For that reason, always one purine has to make a base pair with pyrimidine only. Then only every base pair will occupy the same space, similar kind of space, giving the all double helical structure a uniformity. Okay, and why specifically A with T? A can also make a bond with C also because C is also a pyrimidine. But you can see from the previous slide that A has maximum level of two hydrogen bonds. Okay, G and C they both have three. If I make hydrogen bond A with C and G with T, then all of them will have two hydrogen bonds maximum. So in any case, one hydrogen bond I will lose. To grab this opportunity for the formation of the third hydrogen bond between G and C, we are actually having the bond base pair between A and T and G and C. And what the Chargaff rule is telling that all in a DNA molecule, the total number of purines, that is the summation of adenosine and guanosine, will be equal to the total number of pyrimidines. That means that C plus T. That is, if we summarize the rule, then it will be A plus C equals to G plus, sorry, it will be A plus G will be equals to C plus T. Okay, and the ratio will be ranging from 0.4 to 1.9. So the helix diameter, when the helix will be formed, it will have a 20 nanometer of diameter or 20 angstrom in the diameter. So from here to here, it will be 20 angstrom. Okay, and from one base pair to the other base pair, the difference will be 0.34 nanometer. And in one helical turn, there are 10 base pairs. So if between two base pairs there is a space of 0.34 nanometer, so definitely in a total helix, in a total round of a helix, it will be 3.4 nanometer, 0.34 into 10. Okay, so the whole length of this portion will be 3.4 nanometer. And the pitch of the helix, that means the angle between one base pair to the next base pair will be 36 degree. Okay, So as we know that 10 base pairs are present in a helical turn, so it will make a all 360-degree angle. Major grooves and minor grooves, these two are the very important part of the DNA structure. Whenever two strands are moving antiparallelly, they will form two grooves. I mean, the difference between two strands, this is the difference between two strands. These are known as the minor grooves. I mean, a very small groove, okay? And the difference or the distance between two, this helix, this is known as the major groove. There is a very big groove present over here. I mean, bigger than this one. So this is the major groove, this is the minor groove. These grooves are actually important for stacking or the for binding, tight binding of the protein molecules again. Okay, if the molecule will be very smooth in structure, no grooves are there, then the protein cannot bind very tightly with the DNA molecule. As I told you that several protein molecules has to bind with the DNA molecule or in the RNA molecule, so it should have uniformity as well as it has some grooves. So these are the grooves, major grooves and minor grooves. These are very much important in the DNA structure. Majorly three types of DNA are present in the nature: A DNA, B DNA, and Z DNA. A very slightly present in our body. Z is actually not present. It is, yeah, we can synthesize it in vitro, but in our body, what we have, that is the B DNA, B kind of DNA molecule. Okay, for the structure we have, I have told for the last few minutes, the all the structural features are of B type of DNA molecule. So these three are the major types of DNA molecules. Here are some differences between B, A, and Z form of the DNA. Okay, B and A, they both are the right-handed helix, and Z is the left-handed helix. Base pair, as I told you, that 10 base pairs are present in per turn. In case of A form of DNA, this is the 11, and in case of Z form of DNA, it's the 12. Okay, and a vertical rise per base pair, this is the 3.4 Armstrong, as I told you, and it will make 0.33 Armstrong in base pairs. Here, in case of A form, it is 2.56. In case of Z form, it is 19 Armstrong. Okay, ration per base pair is 36 degree. As I told you, the angle between two base pairs, it is 36 degree, and it will make a whole 360-degree angle when one round will be completed. Okay, in case of A form, it will be 33 degree, and in case of Z form, it will be minus 30 degree. And helical diameter, it is 19 Armstrong, almost equal to 19 point something. Actually, it is almost equal to 20 Armstrong, and this is almost equal for all the form of the DNA. Now we are moving on to the RNA. It is another form structure of the nucleic acid, the second form. It is also a very important thing because RNA actually carries all the information from the DNA to the cell. Okay, ribonucleic acids, as I told you, that if in the 2' position O group is present, then this is known as a ribose sugar. And if the ribose sugar is present in the backbone, then it is known as a ribonucleic acid or RNA molecule. This is the very basic unit of an RNA molecule. Here you can see that this is the 2' position, and here OH group is present. But in case of the DNA, here only H was present, O was absent. Okay, for that reason, that was the deoxyribonucleotide, and it is a ribonucleotide. Rest of the structure is almost similar. It is also going from the 5' to 3', and here also one phosphate group is present. It will make a bond over here and over here also. So it will again go from the 5' to 3' direction. The phosphodiester bond, it is one ester, it is one ester. So one phosphate is making two ester bonds. So altogether, it's the phosphodiester bond clubbing two ribose sugars together. Okay, and this is the nitrogenous base. As I told you earlier also that in case of RNA, four nitrogenous bases again are present. A, G, and C are common with the DNA, but instead of T in RNA, U is present, uracil is present. Okay, so these are some differences between DNA and RNA. As I told you that in case of sugar, in DNA, it is a deoxyribose, oxygen is not present. In case of RNA, it is a ribose, oxygen is present. Okay, these three are common: adenine, guanine, and cytosine. Uracil is present in RNA, and thymine is present in DNA.
Rajat sir, can you hear me? One has unmuted himself or herself. Please mute your audio. We are requesting again and again, please mute your audio.
Okay, I'm calling up Rajat sir to rejoin once more.
Okay.
Hello, 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 hello.
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 Yeah.
Yeah, Thakurji, Professor Thakurji, is there any
Yeah,
problem?
yeah, you got disconnected after the DNA. After that slide, that DNA, RNA, and RNA, the differences between DNA and RNA. Thereafter, you got disconnected. So I think it will
Okay,
be
okay,
better
okay.
if you resume your talk from that slide only.
Yes, yes, yes. Okay, okay. I just made my mobile silent, so I could
Okay.
not hear.
Hmm.
Okay, okay. I'm very much sorry, very much sorry.
No,
So
no, no.
streaming is going on now?
Yeah, streaming is going on.
Okay, okay, okay. So I'm going from that slide only, okay?
Okay.
Yes, one second, give me time. From this slide?
One second. Yeah.
Yes. Okay. So from this slide, if we are resuming our talk that the DNA between difference between DNA and RNA, what happened that DNA can be differentiated with the RNA, that DNA is a double-stranded structure. As I told you, that DNA is a double-stranded double helical structure. Here the difference is you can see that ribose sugar in the sugar molecule, here is no OH group. Okay, here is only the H group. So this is the deoxyribose, and this is the, in case of ribose sugar, there is an OH group present over here. Okay, so this is the ribose sugar. So this is the major difference between DNA and RNA. Here is the H, here is the OH. This is deoxyribose, this is ribose, only ribose. And as I told you that thymine, this is the thymine sugar, new nitrogenous base. This is present in the DNA. And in case of RNA, instead of thymine, there will be the uracil. And rest three groups are present. Rest three nitrogenous bases are present commonly in both DNA and RNA. Adenine, guanine, cytosine. These three are present. Okay. So this is the basic differences. Although there are several differences among the DNA and RNA molecules, but for this case, I am just saying the basic differences between the DNA and RNA. There are some secondary structure of RNA molecule. I am telling that following are the forms of the secondary structures of RNA molecule. Here, the stem and hairpin loop structure can be the major structure, secondary structure of the RNA. In the same, if one RNA chain is going from here and the other RNA chain is going from here, so as I told you that nitrogenous bases are present in the RNA molecule, they can form the they can form the hydrogen bond amongst them to form a stem like structure. And if a single molecule of RNA is forming the stem structure, and in between they cannot have any base pairing among between them, so um, they can form a bulge like this. This is the hairpin loop structure. Hmm. And this is the pseudoknot. If the two different this hairpin loop structure come together to by a single molecule of RNA, then this kind of structure can be formed, which is known as pseudoknot. This is the bulge. If any unpaired bases are present, then they can form a bulge or internal loop kind of structure. And a multi-loop, that means more than one loop if is present or stem is present in one single RNA molecule, then it can form a multi-loop structure. So these are several secondary structures of the RNA molecule. Now we are moving on to the types of RNA molecules. Different types of RNAs are present. One is the messenger RNA, the other one is the ribosomal RNA, and the other one is the transfer RNA. These three are the molecules. I'm telling that this is the major types of RNA molecules. Okay, this is the major. There are many different types of RNA molecules can be present in the cell, but these are the major types of RNA molecules. One is the messenger RNA or mRNA. these actually these have the message from the DNA, and it will give the message to the protein molecules. So these actually carries the message from the DNA. Next is the tRNA. These are actually transfer the message from the mRNA to the protein. Okay, through the incorporating different amino acids. And the ribosomal RNA, third portion of the ribosomal, third type of RNA is the ribosomal RNA, or commonly known as the rRNA. These are the major component, major part of the ribosome. Okay, in addition to the ribosomal protein, these are the major component of the ribosome. So this is the mRNA, different types of mRNA. As I told you, mRNA actually takes the message from the DNA. So in mRNA, in our case, in the eukaryotic cells, mRNA have 5' capping and 3' poly A tailing. In case of prokaryotic mRNA, these are not present. That will be talked by Professor Thakur Chaudhary later on. And uh, this is the UTR portion. UTR means the untranslated region. They cannot translate into the protein. Rest is the translated portion. This portion will be translated into the protein molecule. So actually, according to the mRNA, protein will be synthesized. So the information what DNA is keeping with it, it will be transferred to the protein ultimately. This is the tRNA structure. In the tRNA, it is the transfer RNA, as I told you before also, that in tRNA, it is the two-dimensional structure of a tRNA. It actually transfers the amino acids to one another according to the mRNA information. And in this tRNA structure, four stems are present. This is the acceptor stem. This is the T-shy loop. And a stem. This is the anticodon arm, and this is the DHU arm. Okay. So here one loop, two loop, and three loops are present, and four arms are present in the secondary structure of tRNA. This is the here the T-shy loop. Here shy is one nucleotide present. In the DHU also, DHU one nucleotide, this is present. For that reason, this is known as DHU loop. And this is the anticodon loop. That means when it will bind with the mRNA, the codons. What is codon? Codon is the, three nucleotides get together to form one codon. That codon will be interacted with this portion of the tRNA, and as it is the anti of the codon, for that reason this is known as the anticodon loop. Okay, when the three-dimensional structure will be formed, then it will look like this. This is the English, revert of the English L letter. It looks like. So this is the structure of three-dimensional structure of the tRNA molecule. This is the rRNA molecule, ribosomal RNA. Here, actually, many different types of secondary structures are present. Ribosomal RNA, these are the major component of the ribosome, as I told you. Apart from the protein, these are the major components in the ribosome, and they can have different types of secondary structures. Now I am coming to the next thing, which is the last macromolecule, last type of the macromolecule, which is the fatty acid. Here, as I told you, that the fatty acids is a carboxylic acid with a long aliphatic chain, and they are one of the major components of the lipid molecule along with the glycerol. In the lipid molecule, I told you that along with the glycerol, two fatty acid chains are interacted together. Okay, so in the OH groups present in the glycerol, two fatty acid chains are present to form that tail groups. Glycerol will make the head group, and two fatty acids will make the tail group of a lipid, phospholipids. So fatty acids, depending on their bond between the carbon and carbon, fatty acids can be differentiated into two different categories. One is the saturated, the other one is the unsaturated fatty acid. In the saturated fatty acid, what will be there? That in the saturated fatty acids, no double bonds are present between the carbon and carbon atom. Okay, as I told, as you all know, that what is the unsaturation? In the unsaturation, one will be the sigma bond, the other one will be the pi bond, and the pi bonds are pi electrons are hybridized. They're not hybridized, so they can form another bond with anyone else. So as they're unsaturated in nature, so these are the unsaturated fatty acids. If double bonds are present between the carbon and carbon, it will be the unsaturated fatty acids. And if no double bonds are there, it will be the saturated fatty acid. So if the unsaturated fatty acids are there, it will be two types of conformation. One is the cis conformation, the other one is the trans conformation. In the cis conformation, what will be there? The double bond, if the similar type of groups are present in the same side of the double bond, then it will be the cis conformation. And if the similar kind of groups are present in the opposite direction of the double bond, then it will be the trans conformation. So this is the trans fatty acid, this is the cis fatty acid. This will be synthesized. This will be the two types of the unsaturated fatty acids. And in depending on the number of the carbon chains, how many number of carbon are present in the chain, depending on that, the fatty acid can be of two types. One is the odd chain fatty acid, the other one is the even chain fatty acid. Odd chain means if the odd number of carbons are present in the fatty acid, then it will be the odd chain fatty acid. And if the even number of carbons are present in the fatty acid, then it will be the even chain fatty acid. Okay, as I'm showing you that heptadecanoic acid, this is an odd chain fatty acid, 17 number of carbons are present in the chain, and this is stearic acid, this is the 18 carbon chain, that means 18 number of carbons are present in the chain, so this is an even chain fatty acids. Okay, so I'm now at the end of my presentation. Is the energy production, main pathway of energy production. As I am telling you, as I told you, that four different macromolecules we are discussing about today: nucleic acid, lipids, protein, and carbohydrates. And main thrust of all these molecules are to do the work and to make the energy for that work. Okay, so this is the central pathway for the energy production. Sugar, all the sugar molecules will be converted into glucose, either glucose or later on any intermediate stage. The glucose will be converted into pyruvate by the pathway of glycolysis. Pyruvate will be converted into acetyl CoA. Acetyl CoA will get entered into the TCA cycle, and from the TCA cycle, NADH and FADH2 will be generated, and they will go for the oxidative phosphorylation for the synthesis of ATP. Okay, all the fatty acids, if it will be, degraded, it will form the acetyl CoA. Again, it will come to the citric acid cycle, and the same pathway will be followed. And the amino acids, the different 20 types of amino acids may also be converted into the different parts, different intermediates of this PCA cycle, and they will get entered into the PCA cycle, and they will also follow the same pathway to for synthesis the ATP. Okay. If it is an oxygenic condition, then pyruvate can form the lactate and ethanol. They can have some NADH molecules also, so that they can be again get entered into oxidative phosphorylation. So this is the central pathway for the energy production. You can see that carbohydrate, all the sugar molecules, proteins, all the amino acids, and lipids, the fatty acids, these three can get entered into the energy production. That means they three can contribute themselves for the production of the energy in the cell. You cannot see nucleic acid here because, as I told you, that nucleic acid is a supervisor, central supervisor. They will just look after whether this pathway is going smoothly, running smoothly or not. Okay, so nucleic acid will not directly involved in the energy production, but yes, nucleic acid will take care about the pathway whether it is going smoothly or not. So this is the end of my lecture today. Thank you very much for being with us. Thank you for listening to me, and I'm very much sorry. In between, I got disconnected, and sometimes you have to stay without listening my lecture. But we cannot do anything. We have to bear these kind of problems in this virtual platform. So I'm done.
Thank you very much, Professor Rajat sir. Thank you very much for the wonderful talk. I'm sure the students have really learned a lot. They have really brushed up their basics on carbohydrates, lipids, proteins, and nucleic acids. Coincidentally, I would like to share one fact with you all that
Yeah.
today is the beginning of the National Nutrition Month, which will be observed from the 1st of September till the 30th of September. And as we all know, carbohydrates, lipids, as well as proteins, they are an integral part of our nutrition. So there could have been no other befitting topic for today's talk than the basic biological macromolecules. So Rajat sir, thank you for speaking on this topic, and we all have gained a lot. Thank you so much.
Thank you. Thanks a lot.
Uh, as I said, we have a question in
Yes,
our chat box.
yes,
The question is from, once again, the question is from Bhakti Thenge. Where are
mhm.
the Z form of DNA is present?
Yes. Very good question. Actually, I told this also that Z form of the DNA is not actually present in biological system. We can form, this is a probability to form the DNA structure. Okay. This we can form in vitro. Okay, so the structure what it has, it not suits with our biological system. As I told you that in the DNA, several protein molecules should bind, several activities should be done, and that thing cannot be happen with the ZDNA, Z form of DNA. For that reason, that is a probable structure, that is a discovered and established structure that can be present in nature, but unfortunately till now it is not discovered that any one of the living system has this kind of DNA. This can be synthesized in vitro. Okay.
Thank you, Rajat
Oh,
sir.
okay, sir. Thank you, sir.
Okay.
So
Welcome,
I
welcome.
would like to thank all the audience also for participating in this talk and for waiting patiently, even despite of the technical issue which we faced.
Yes.
And we
Mhm.
will all meet tomorrow with Professor Shomshubrat Thakur Chaudhary's talk on a basic insight of the central dogma of molecular biology. Thank you, all of you. See you all tomorrow. Thank you, Rajat sir, very much for the wonderful talk.
Thank you.
Thank
Thank
you.
you,
Thanks
everybody.
a lot.
Thank you,
Thanks a lot.
everybody. Okay.
Have a nice day.